Hello and welcome everyone to the Carbohydrate Chronicles, a podcast about glycoscience and glycoscientists. In this show, via a series of interviews, we explore the world of carbohydrate science and innovation from a personal point of view. Your host today is Tamás Hyde, a member of CarboHyde scientific team. Glycoscience is the study of carbohydrates or sugars and their roles in biology. Cyclodextrins are a very peculiar type of carbohydrates, ring-shaped molecules composed of glucose units. The traditional uses of cyclodextrins within the pharma industry are small molecule solubilization and formulation, yet there are novel fields emerging like protein stabilization, gene delivery, using them as therapeutic agents, or exploiting their potential in biotechnology. Overall, cyclodextrins have important implications for human health and disease, making it a very relevant topic in modern research. Today, we have a very special guest with us, Dr. Milo Malanga, a renowned scientist in the field of cyclodextrin and carbohydrate synthesis, whom I have the pleasure to know for over a decade. Welcome, Milo. It's nice to have you on the show.
Ciao, Tomasz. It's very nice to be here. Hello, everyone, and good afternoon.
By way of introduction, Milo is an author of over 50 scientific publications and several patents despite his young age, and one of the most well-connected researchers in this field. His background is in medicinal chemistry, and he focuses on creating novel cyclodextrins for innovative applications. Currently, he's also the principal investigator from CarboHyde side in the Genegut project, a Horizon Europe grant focusing on discovering novel drug delivery systems for oral gene delivery against Crohn's disease. Milo, I'm really interested to learn how you ended up in science. I know that you studied medicinal and pharmaceutical chemistry in Bologna, yet what made you choose this path? Was it clear and straight right from your childhood?
Not really, not from the childhood at all. Okay, I fall in love with medicinal chemistry during the period of the university. I had the pleasure to be a student of a wonderful professor, Giovanni Gottarelli, who teach me organic chemistry at that period, and I fall in love with the subject. I was hooked at this subject at that period, and then I decided that I want to do synthesis and continue in that way. I was always curious of how reaction can go and where a new reaction can go. So it was at that time, during the period of the university, in particular during the thesis period, that I got hooked with the research.
How about earlier periods? I mean, when you were a smaller kid, were you also drawn to natural sciences, not, I don't know, automobiles or engineering or whatever? Was it always chemistry and biology that you admired?
No, absolutely no. I was in love with mathematics. I mean, I'm still in love. I mean, I was far really. I always been in love with mathematics, and I always still thinking that okay, as a second choice, if it's not organic chemistry, I would love to teach mathematics, and I'm still practicing and studying by myself sometime when I have free time, and it was lovely for me. I mean, it's intriguing and creative. And when I was a child, I don't know if it's connected to this one. I always love to do puzzle. I'm still doing it, and I'm still in love with them. Doing puzzle in my free time, it relaxes me, and it's made me curious. I mean, how to match piece by piece in the correct position, spatial position, and piece.
That's funny. That's something that we have in common. I also have the second fascination towards mathematics. So if I weren't a pharmacist, probably I would have been an economist or something like that, where I could practice mathematics too.
That's nice to hear. I didn't know, Thomas. That's okay. We have this one in common. It's very nice to hear.
So when you got deeper into synthesis during the university, was it carbohydrates from the very beginning?
Not carbohydrate, let's say partially. We were working with nucleic acid at that time, but in a peculiar way. We were studying self-assembly of nucleic acid, and I was really curious about the supramolecular interaction of molecule. Of course, we end up with carbohydrate because being a pharmaceutical-driven university and faculty, we were dealing with a drug based on carbohydrate at that time, a mix of carbohydrate and cyclobate. We were studying utilizing antiherpetic drug in that period, and carbohydrate were part, of course, of the molecule of interest. For the first time, I came in touch with cyclodextrin with the period of the PhD. I was already here in Hungary, in Budapest, when I started with that.
I was just about to ask that. So, what was the first time that you came across cyclodextrins? What's the first memory with them?
It was late. I mean, the period of the PhD. The very first time, it was one of my last exam decades ago at the university, where a professor dealing with mass spectrometry asked me to analyze a paper where cyclodextrin had been used as a chiral selector for specific molecule. That was the first time with the research article. But from the practical point of view, just during the period of the PhD. We were involved with several countries in Europe in this cyclone grant from the European Community, and based on cyclodextrin for development of a new anti-cancer drug. And at that time, okay, I started to work heavily with cyclodextrin, and I fell in love with the molecule, which are extremely challenging to modify to with the infinite property, I would dare to say.
I understand that for the purpose of this educational grant, you left Italy. Was it always the plan to do some studies and work abroad, or it just came by chance?
No, that one was one of my firm conviction. I always said, despite I was proposed to stay in Italy to do the PhD, that if I was deciding to do the PhD, it must be abroad. I wanted to have an experience not in Italy, abroad, to learning from other professor in other environment, in other lab, and it was a beautiful experience. It was the right choice for me because I had a lot of experience in other laboratories, taking the best from laboratories around Europe during the period of the PhD. It was a crazy period for the traveling, really. I was not expecting so much moving. Once it happened, okay, that really I didn't know where I was anymore. I had all my stuff, I don't know, part in Spain, part in Italy, part in Budapest, and I couldn't remember neither where the clothes were. So it was a crazy and exciting period, that one of the PhD, that I would recommend to everyone.
How about your internal drivers and fascinations? So it's clear that you're in love with chemistry, but what is the most joyful part for you in it?
What I enjoy most, the unpredictability. No matter what, it's like be a bit. I don't know. It's always painful to read a reaction because you don't know what to expect. No matter how much you can find data in the literature, how much the reaction is described, it's always something that can I don't know, can surprise you. I mean, as I always say, okay, that as a chemist you have to be ready for failure, because that is practically is a normal way. When you do a reaction, 90% of the cases the reaction are unsuccessful. That is what is stimulating. Of course, on period you can get down, but in my case, it's stimulating because you have to have always be ready with plan A, B, C, D till Z. I mean, there is always the need of more option because synthetic chemistry in general and carbohydrate chemistry is so challenging and unpredictable, and this is the beauty of it, that there is a lot to do, and it's super stimulating, and it drives my curiosity. I would say that really curiosity is what is a driving force for doing chemistry for me.
You had part of your studies and research in an academic environment, experiencing works at multiple universities, and you had also the chance to work in industrial environment. Is industry your final pick, and why is industry your final pick if it is?
I would say yes, industry and the private sector is my final pick. I love it. It's different from academy, but I would say that I like an industrial environment where there is a possibility to train students. This one is something that I love in the academy. I always thought to be a teacher in my after the university. I was thinking, okay, I want to be a teacher at the university. But once I get in contact with the private sector, I fall in love. Especially, I had the opportunity to have a lot of freedom in during my PhD, and I embrace all the experience in the private sector, which is dealing with deadline, with GMP production, or very in a very rigorous manner. Still, I love from the academy the fact that you have the possibility to be in contact with students. And at CarboHyde, for example, we are creating an environment where is a mix of the two. I love to spend time with the student in the lab to train them, if possible, to establish new collaboration. From the other side, industrial sector is my field because of the rigorosity of the approach and of the fact that you have a strict deadline and okay, you have to deliver on time. You are a bit under pressure, and it's always drives the best out of me. At least I hope. I don't know if my colleague will agree with me about this one, but this is what I truly hope.
So you want a little bit of both actually in your
Yeah,
everyday work.
yes, that's correct. To merge the two fields, academy and private sector, even more.
Nowadays, you are leading the scientific operations of CarboHyde, which is, of course, to some extent a different role than being beside the bench running with reactions. How do you cope with transition from a chemist to a leader? Is it easy for you?
Sometimes it's challenging, let's say, to put it in a nice way. As I mentioned, you know very well, Thomas, I love staying in the lab and running reaction. That is what really my true love. To transition to a leader position and be a chief scientific officer, it was not easy for me, honestly, and I'm still coping with this new position. But I accepted the challenging. There are a lot of things that I'm learning, and I'm challenging myself constantly in sectors that are not, or fields that are not, they are totally new for me. But I accepted the challenges, so I'm adapting at the moment. But I found the beauty also of business development, interacting more, let's say, digitally with person rather than physically, and which is, you know, I'm Italian. I love physical content and to meet a person. I mean, when I don't know, be digital too much is, I don't know, it could be a strong compromise for me sometimes. But I'm coping with it. I'm up to the challenge.
Yeah, I guess life is an endless learning process, so we to improve ourselves day by day.
Totally
How,
agree about this one.
how about interests beyond cyclodextrins? So, what else are you interested in? What kind of, I don't know, molecules, chemistry?
Me, in general, not only psychology, I love carbohydrate because, in my opinion, are the most difficult molecule to modify synthetically, and they have infinite potentiality. In particular, one of our also long-term project is to develop API based on carbohydrate. It doesn't matter, doesn't matter which disease. It's is intriguing for me the idea to design and use carbohydrate for these purposes. There is so much that is unknown about chemistry of carbohydrate in the body, in the human body, that in my opinion, a lot of potentiality or a lot of unknown in this one, and a lot of parts are still undisclosed. This one is really stimulating for me. So cyclodextrin, of course, will be always part of my research, and they have a special part in my heart. But in general, carbohydrates are the molecule that I love to deal with for any purpose, not only strictly related to the pharma field. I love also to think about carbohydrate as a material science, as a material science tool to create something so specific material for dedicated purposes. So to enlarge the vision, I love this idea to apply in material science.
Switching the topic a bit, what do you do when you're not in the lab and not working? What makes you happy when you switch off?
What makes me happy, I think the person that are close to me, they know, okay, there are two things that mainly I love doing. One is reading comics. That is my second option if I'm not reading a chemistry article. The other one is go to the cinema. I love movie. I'm really a devoted lover of cinema, and I was struggling really a lot during the period of the COVID. There was not the possibility to go to the cinema. So these two things, yeah, are principal my hobbies.
Getting back to professional parts and challenges and your closer interest, the cyclodextrins, which is the application area of cyclodextrins that you're currently most excited about or you think holds the most promise?
Yeah, what I'm most excited, maybe I'm repeating myself, is to design cyclodextrin as a API for pharmaceutical use. in particular to develop an API based on cyclodextrin. And we are still at the beginning because really they have infinite potential as a molecule to be utilized. Topic that fascinating me about cyclodextrin. Besides all the other use, if I have to pick one, I pick this one. No doubt about that.
Certainly, using cyclodextrins as active ingredients is an upcoming and emerging field with a lot of new biotech startups working on it, apart from CarboHyde as well. How about drug delivery? Where do you see the best potential in using cyclodextrins as drug delivery tools?
That is a very interesting question, and I think that cyclodextrin have a very high potential in drug delivery. CarboHyde is involved in Genegut project, and as you know, cyclodextrin have a pivotal role in this project because we are trying to use them as delivery for RNA for treat Crohn disease, and I think that this molecule fitting perfectly the task as nowadays even more sophisticated compound are necessary for doing the task. As we know very well from the experience with the previous vaccine related to COVID, the vehicle are as much demanding to be prepared than the active, and are even more sophisticated, and there is really necessity for having alternative to the current one that are utilized, and cyclodextrin can fit perfectly the job, can do perfectly the job. We need, of course, companies and personnel able to modify them ad hoc for the specific target.
Can you name a specific project or an accomplishment that you're particularly proud of in your career so far?
Yes, I think I can. There are two projects that I'm really proud of. One project is for sure the synthesis of all the sugar madex impurities that we developed during the last 5-6 years. This one I'm really proud of it. It was really stimulating and really beautiful to learn how many derivatives you can create starting from one single molecule. And the other one is the collaboration that we still have with the Cyclarity for the development of cyclodextrin dimer for treating atherosclerosis, in particular to interact with selectively with ketocholesterol. That is something, a project that I still has a particular place in my heart. We were involved from the beginning. We are still collaborating with them. We have promising results, and I love collaborating with them.
By the way, among the series of interviews, we also have one with Cyclarity's CEO, so you can learn more about that development in another episode. How about failures, Milo? So you were very enthusiastic about successes. Do you have any project that you can name where you, I don't know, went the wrong way and didn't arrive to a success, or at least that was a good lesson?
a lot of if you think about, as I mentioned before, synthetically speaking, a chemist has to be ready for failure, because that is the common way when you run reaction in the lab. That one, it doesn't have to drive you down, but it should be a stimulus to do better. But an entire project, but yeah, I had experience with that one, and I give detail in general. I mean, we were developing an API, and at a certain point, we were closing to the end of the project, and we realized that the analytical method that was at the base of the analysis of the API was totally wrong. So the project went south. And you can imagine I was really angry and upset, but what to do? This one is a lesson that you learn, and it was really profitable for me for the future, that it increased even more the consciousness that without powerful analytical method, doesn't matter, a synthetic chemist can be lost, especially in the pharma field. If you have to develop API, there is a very deep connection with the analytical team, which is not so obvious, believe me, because sometimes organic chemists and analysts, they speak different language. Is really sometimes challenging to communicate, but this is the basis for a good process. This one I learned really in the harsh way, in the hard way, let's say.
Yeah, we all learn the best lessons probably that way. So I understand that you see the future, at least your fascination and drivers are to create new cyclodextrins as active ingredients. At least that's one of the core projects that you're working on now. For this vision to become reality, what do you think is the biggest challenge that you need to overcome synthetically or medicinal chemistry wise?
Synthetically, there are still a lot of challenging, no matter how much researcher who I admire putting a lot of effort in developing new regioselective synthesis or regiospecific, there is still a lot of things to do. Cyclodextrins are really challenging molecule because of the multiple functionalities, and there is really a large room of improvement. And still, I think that this one is one of the main drawback. The synthetic approach, especially when it's involved multi-step synthesis, is not straightforward to be translated to the industry. There is a lot of work to do in the simplification of the mostly academic approach, which are quite elaborated and demanding. Simplification from the practical point of view of organic chemistry, and time. You need time, and of course the right funding from the industry, and to increase the interest toward cyclodextrin and carbohydrate, because the potentiality are infinite for this molecule, and we are at the beginning. We need time and patience and always curiosity. That one for sure.
Yeah, that's a good line. I like that one. Again, let's drive away a bit from science. Question that we have always on the show that I'm gonna raise. If you could have dinner with a historical figure or celebrity, dead or alive, who would it be and why?
Okay, the historical figure that dead, I guess. I mean, I would choose maybe Leonardo da Vinci, probably.
Ah,
Must he was a genius. It could be a very stimulating discussion to have about painting or whatever art, or I don't know, invention.
for science.
About
Yeah.
yeah, science for sure. About celebrity, I would choose for sure a movie director. I don't have doubt. If I have to choose one of alive, I would choose Clint Eastwood, one of my
Oh,
favorite director and actor. I mean, he's nowadays how old is 90 years old. He would have a lot of story to tell, and really, I love his movie. It was funny also when he was an actor. I mean, during the Western movie, so it could be a lot of fun to have a dinner with him.
so you would pick from your hobbies, not from your work, not a scientist or something like that. That's
Don't know this one. I mean, no, not Einstein for sure.
So you mentioned that you have a very great need and necessity to be involved in education and teaching of the next generations. What would be your advice for young scientists who start their careers in the carbohydrate field or just outside in science? What's your main takeaway as an experienced researcher?
I would have just a really basic suggestion. My main one would be stay curious and be critical of what your experience, what is your discovery, and follow your passion, no matter how much rhetorical this sentence can be. I always follow my dreams and my passion, and I end up in beautiful place. I had beautiful experience, and I would suggest to do the same. Don't give up ever, and follow your dreams.
Thank you very much, Milo. It was really great to have you on the show, and I really enjoyed the conversation with you. So thank you for your time.
Thanks to you, Thomas. It was a pleasure for me as well. Have a nice day.
I hope you enjoyed the most recent episode of Carbohydrate Chronicles today, delivered to you by CarboHyde. We talked with Milo Malanga, CSO of CarboHyde, and learn how passion and persistence pays off and could help one develop a unique career in this niche area. As Milo sees, and of course I can only echo his view, there is much more to expect from cyclodextrins. We can only encourage young and not so young researchers to dive into this area and explore fascinating novel applications in drug delivery or active ingredient development as the possibilities are truly endless. And I'm sure you can count on Milo's support to overcome any challenges that you may have. I'd like to thank Milo for accepting kindly our invitation and allowing us to peek into his professional and personal life, drivers and messages. And thank you all for listening for this interview. Next week, in the next episode of Carbohydrate Chronicles, we'll talk with Anne Imberti, a pioneer in structural glycobiology from CNRS France, with main interests in biologically active oligosaccharides and their interaction with lectins and glycosyltransferases. Your host was Tomas Shahida from CarboHyde, and I hope you'll join me in our next section too. See you in 1 week. Goodbye.